Last time we'd gotten to the point where we had laid out the polymeric proteins of tubulin and forming microtubules, and globular actin self-assembling into a filamentous actin. What we're going to do is we're going to recapitulate that a little bit, but what I want to emphasize is the idea of microtubules and actin as being substrates upon which we can transport vesicles. And we'll talk about this as microtubule and actin-based transport. The long-distance form of transport is going to be primarily on microtubules. And the long-distance form of the transport is going to depend on two major motors. One motor is called Kinesin, and the other is called dynein. Now, one of the important characteristics of Kinesin is that it's always a plus N directed motor. And you remember from last time when we laid out the microtubule as a polymer, we said that it had an endedness. It had an assembly end and it had a disassembly end. And the assembly end we said was the plus end, and the disassembly end we said was the negative end. Now in biology, in reality, those microtubules are going to be stabilized. So we have a fairly stable structure upon which we can move vesicles. But the motors that are going to march along the microtubules are either going to be a plus N directed motor called Kinesin or a negative N directed motor called dynein. So we'll lay out the microtubules. We'll orient the microtubules from the negative end to the plus end, and then describe how these motors, these molecular motors, can bind to the microtubule and move vesicles along those substrates. We'll see that not only are microtubules the basis of transport, but there's also another substrate along which we can move things, and this is going to be filamentous actin. But the filamentous actin is going to use a different motor. The motor that's going to move on filamentous actin will be myosin. But what we're going to see in all cases, whether we're dealing with kinesin, dynein, or myosin, in all cases, these are ATPases. What they do is they convert adenosine triphosphate to adenosine diphosphate. And one of the take-home messages from today's discussion is going to be that all three of these motors have the capacity to take chemical energy stored as adenosine triphosphate and convert that into mechanical movement. And it's that mechanical movement then that's going to be used to direct the vesicles one place or another inside the cell. And that direction of the movement is determined by the substrate. The plus end and minus end of the microtubules, the plus end and the minus end of the actin filaments. And then once we move from there, we'll talk about the central dogma. Again, DNA stores information as a sequence of bases in a gene. RNA is an intermediate between DNA and protein, and it also stores information as a sequence of bases in the RNA transcript. And then the final step is translation, where information in the gene-based sequence is expressed as an amino acid sequence. And we'll spend some time on that. And then to understand the central dogma, we'll begin with some nuts and bolts about DNA structure, emphasizing that deoxyribose is a 5-carbon sugar. And the 5-carbon sugar can be linked into a great chain through phosphotoester bonds. So we can take these deoxyribose sugars, link them together through phosphotoester bonds, and make a great long polymer. But then the nitrogenous bases are also covalently linked to the deoxyribose. And there's a covalent linkage of adenine, guanine, cytosine, and thymine. Adenine and guanine are said to be purines. Cytosine and thymine are said to be pyrimidines. And we'll talk a little bit about what this means. And then when we put the DNA molecule together, we'll find that the two strands are always linked by covalent bonds from the number 5 to the number 3 carbon on the sugars. But the nitrogenous bases are linked one to the next by hydrogen bonds. And adenine always binds to thymine, and guanine always binds to cytosine. And we'll work our way through that. But let's begin with this description of microtubules as polymers. of tubulin. Now, we talked a little bit about this last time where we said that tubulin was in itself a dimer composed of alpha and beta tubulin. But each of... Time's up, yeah. Well, time's racing. Time's up. Okay, so the alpha and beta tubulin now can self-assemble such that the beta is bonded to the alpha, the beta is bonded to the alpha, and the beta is bonded to the alpha. And so this now extends into this great long linkage. So we can now link one of these one to the next. And there's an end to which it assembles, said to be the plus end, and an end from which it disassembles, said to be the minus end. Now, in addition to this ability to link end to end, these tubulins have the ability to link side to side. They're always oriented alpha beta, alpha beta, alpha beta. But now they're linked side to side, and they're usually offset by a little bit. Now, the logical conclusion of this is if you did this, you'd have this great sheet of tubulins all linked together. And that doesn't occur. What happens is that there's a slight curvature from one strand to the next. So that when you look at these side by side, when we change the orientation, rather than looking at these edge-on, we now turn it 90 degrees. When we do that, we find that there are 13 of these. There are 13 alpha beta tubulins oriented in this circle. And this provides now a 25 nanometer tubule. It's 25 nanometers in diameter. Now, it can be enormously long. These can be up to 10 micrometers or more in length. So they'll be 1,000 times longer than they are wide. And so this microtubule will extend out toward you, toward the plus end, and extend behind the board toward the negative end for hundreds of meters at this sort of dimension. But no matter where we look at these, they always have a negative end and a positive end. Now, depending upon the function of the microtubules, some of these are stabilized and some are not. We're going to deal only with those that are stabilized. We're going to say that the microtubule has been generated. It's been generated by self-assembly of these alpha-beta tubulins. And it will have a negative end and it will have a positive end, but that's stable. We're not going to think about the treadmilling. We're not going to think about how they come and go. The next thing I want to do now is I want to place those microtubules in a kind of context. And I'll place them in the kind of context where we can describe microtubules. In neurons. I've deliberately used the neuron because it's an unusual cell. The neuron is a highly asymmetric cell. And if we would draw a neuron, one of the characteristics of neuron, and we'll emphasize this later in the course, is to say that this region up here, this bulbous section, is said to be the neuronal cell body, sometimes known as the soma. It's characterized by many of the structures we talked about last week. There's a nucleus. Associated with the nucleus is endoplasmic reticulum. There will be the Golgi apparatus. This is the Golgi. And from the Golgi, will generate vesicles. Now, one of the peculiarities of neurons is that all protein synthesis is confined to the neuronal cell body. So that when we look at the position of the ribosomes, some ribosomes are attached to the ER, as roughly are, and some are out here in the cytoplasm. But whether we're describing free ribosomes or rough endoplasmic reticulum, those that are attached to the ER, those are always confined to the cell body. Now, why this becomes important is that these neurons can be extremely asymmetric. They can have neuronal cell bodies that have diameters in the neighborhood of 30, and in a big neuron, 50 micrometers in diameter. But the axon itself, in some extreme cases, can be greater than 1 meter. So this sets up a kind of logistical problem for the neuron. All of its factories to make protein are relegated to the neuronal cell body. But yet those proteins that are being generated here in this region of the neuron have to be used all along the length of the axon, and particularly down here in the region of the neuron known as the output region. So again, all proteins are made here, and yet they have to be used at some region far, far away. So the logistical question is, how do we manage to get proteins that are synthesized here in the cell body out to these faraway reaches? The answer to that has to do with the microtubules. So the microtubules, if we look along the length of the axon, all along the length of the axon are these microtubules. So these green lines that I'm drawing in here are the microtubules. So wherever we look along the length of the axon, we see these microtubules. What's more is those microtubules in the axon always are oriented in one direction. They're oriented in such a way that the negative end is closer to the cell body. So we can spell that out and say the negative end of microtubules in axons is closer to the cell body than the positive end. So that this end of the microtubules will always be the positive end. So we've described a kind of orientation among the microtubules. We said because of the way the tubulin adds, we have a plus end and a minus end. And now we've added the caveat here is we have stabilized these microtubules. Turns out, to get into the weeds, at this end, this negative end of the microtubule, is something known as a microtubule organizing center. And the microtubules will actually be generated, they will polymerize from this site. But that's stable, it doesn't change. And even in these microtubules and neurons, the plus end is stable, so you can't add new tubulin to it. So we have a fixed structure, but always oriented in this way, the negative end close to the cell body and the positive end farther away. Now, the next character in this play is the vesicles that were generated up here from the Golgi apparatus. These are the vesicles, the cargo-carrying containers that contain the proteins that we had produced from the rough endoplasmic reticulum and through the Golgi stacks. So now we have all these vesicles. And the next question, of course, is if we've produced the proteins, loaded them into the vesicles, how do we now manage to get these vesicles to move along the axon? How do we distribute the proteins generated in the factories here to the process that may be as much as a meter or more away? To answer that question, we need to describe here. What we want to describe here is the structure of the molecular motor kinesin. I'm going to put this in parentheses because it's so important, we'll come back to it. Kinesin is an ATPase. It converts ATP to ADP and PI. Now, in crude terms, the structure of the kinesin molecule is a dimer. It contains these alpha helical regions that then form a kind of foot. Now, I've separated the two alpha helical regions one from another. In biology, these two are intertwined. They basically wrap one around the other. And then at this end of the molecule up here, there are light chains. The function of the light chains is to bind the kinesin to molecules in vesicles. So there'll be transmembrane proteins in the vesicles. So again, here's our blue vesicle. There are probably transmembrane proteins here, which will bind to the light chains. The foot of the molecule down here is the ATPase. It's the ATPase and tubulin binding region. So this divot or arch that I put in the foot of the kinesin molecule, this site here, is where ATP binds. And the binding of ATP at these sites rapidly takes ATP and converts it to adenosine diphosphate, ADP, plus inorganic phosphate, sometimes known as pyrophosphate. So for the most part, these feet of the kinesin molecule contain ADP and some inorganic phosphate. Now, when the feet of the molecule, when the feet of the kinesin molecule are in this state of ADP and PI, when they have ADP and PI in that active site, At that point, you can imagine these feet to be sticky. And it's under these circumstances that the ADP and PI, the Kinesin molecule, let's do it this way, it's under these circumstances that the feet, because they're sticky, because the ADP and PI will, in fact, stick to the alpha-beta subunits. So you can imagine this vesicle with its kinesin molecule, or several molecules probably attached, is sort of just vibrating around, wandering around. And when it comes close enough to one of these microtubules, then the stickiness of the feet with the microtubules will now allow the kinesin to stick to the microtubule. But that's not the end of the story. At that point, once they're stuck, and in fact, more accurately, I should draw this as this guy hanging out in space a little bit and this one attached. Then what will happen is that at this site, ADP and PI leave. And it's now replaced by a molecule of ATP. It appears that when the ATP molecule binds to this foot, it changes the configuration of this region in here, where this section in here, called the linker region, if this trailing foot has ADP and PI, when this leading foot binds ATP, it zippers together this linker region. When this two parts of the molecule zipper together, this foot steps forward. See if I can put this into sort of an anthropomorphic sense. So if you imagine me as the kinesin molecule, and the light chains are my hands, and I'm holding onto a vesicle over my head. Now, my feet, the soles of my shoes, have ADP and PI on them. And so because there's ADP and PI on the soles of my shoes, they're kind of sticky. So I'm just sort of dancing around, wandering aimlessly until I run into a microtubule. And when I run into a microtubule, my right foot sticks. And then what will happen is that when my right foot sticks to the microtubule, the ADP and PI leave. So you can imagine the ADP and PI leaving my foot, and in comes an ATP. There's like 100 millimolar ATP, so there's plenty of it. It comes in and binds to my right foot. When the ATP binds to my right foot, it basically stitches together my ankles in such a way that I step one step forward. ADP and PI are on my left foot, so they stick. Then when it sticks, the ADP and PI leave, ATP comes in, and when ATP binds to my left foot, my right foot steps over my left. When my right foot binds, because it has ADP and PI on it, the ADP and PI leave, it binds ATP, and my left foot steps forward. So every time we bring an ATP into this site, the trailing foot comes over. And every time it does that, it binds to the next tubulin monomer. So this step from the center to center distance on the tubulins is 8 nanometers. So that means every time this ATP binds to the every time the ATP binds to the leading kinesin foot, it pulls the next one forward and it makes a step of 8 nanometers. It does this at the rate of about 20 per second. I'll let you do the numbers, but this is every 50 milliseconds. So every 50 milliseconds, this thing is stepping one over the other. It's really lifting them up and putting them down. It's moving right along. but the steps are tiny. So in aggregate, when we calculate the movement of the vesicle along the microtubules, vesicles move at a rate of about 200 to 400 millimeters per day. This doesn't seem very fast or very far, 0.2 of a centimeter in a whole day. But remember, this 0.2 of a centimeter, this 200 millimeter transport is derived from individual 8 nanometer steps. That's a lot of steps. It's a lot of ATP. And basically, you're cleaving a lot of ATP, allowing the foot of this molecule by converting ATP to ADP and PI and replacing the ATP and allowing this to step one over the other. You're now dragging these vesicles along the microtubules at a rate of about 200 millimeters a day. Each step is 8 nanometers, 20 per second. So that's how we move things inside the cell. We're generating these vesicles here in the neuronal cell body, attaching them to the motor, and then attaching the motor to the microtubule and moving the vesicles in one direction along the microtubules. The unidirectional nature of the movement derives from the simple fact that all the microtubules are oriented in one way, always with the plus end away from the cell body. Kinesin then, the upper one, Kinesin is a plus end directed motor. And the upshot of the orientation of the microtubules is that we can generate proteins, put them in vesicles in the cell body, and move them from the cell body toward the terminal. But there's another character that we introduced in the beginning. And that is, let's come back here, that the counterpart for kinesin is dynein. So dynein is the kinesin molecule's counterpart. Dynein, too, is an ATPase. So we can say that dynein is a negative end directed motor. But it too is an ATPase. And it too cleaves ATP to ADP, an inorganic phosphate. And in the process generates one step, which I'm pretty sure is 8 nanometers. Now, if you get into the weeds on these molecules, you find that dynein is a far more complicated molecule than kinesin. It's a much bigger molecule. Even just the motor part of it is around 500, that molecular weight of about 500,000, 500 kilodaltons. So the motor part's big. It's one of the biggest proteins in all of the body. And not only that, but unlike the kinesin, it has a whole raft of regulatory proteins. So you end up with this gigantic molecule that's important for transport. But for our purposes, what I want you to recognize is that the distinction between dynein and kinesin is simply that the dynein is a negative end-directed motor, and like kinesin, it's an ATPase. Again, converting chemical energy to mechanical energy, and probably do it in a very similar fashion. But what it tells us then is that if we go back to this model here of the microtubules, we can imagine generating vesicles down here, and often these vesicles will contain growth factors. These vesicles now can bind to molecules of dynein. Now, if the dynein bound to the vesicle, once the vesicle and the dynein bind to the microtubules, because this is a negative end-directed motor, this vesicle is destined to move in this direction toward the cell body. And in fact, in many cases, growth factors must be delivered from the terminal regions back to the nucleus so that those growth factors can affect gene transcription in such a way that they suppress the death machinery of the cell. But be that as it may, the point here is that we have two transport systems on one road, the road being the microtubule, and the transport systems being differing because of the nature of the dynein being a negative end-directed motor and the kinesin being a positive end-directed motor, and the fact that the microtubules are always oriented in one direction. Yes?
Is it possible for a dynein and a kinesin to walk on the same microtubule, considering they're like...
So this great question. So there is this question. If you have these 28 nanometer diameter tubes, or 25 nanometer tubes, and you have 50 nanometer or more vesicles attached to them, why don't things run into one another? And perhaps they do at times. But at the same time, it looks like an individual microtubule can accommodate several kinesins and several dyneins at the same time, and traffic will be moving in opposite directions. In fact, this was sort of a surprise. I'm old enough to remember, and I told the story in the first section, when this was first sorted out. And the way it was done is that these experiments were almost all done, the early ones were done at the Marine Biological Laboratory in Woods Hole. And the reason they were done there, two factors. One is the fellow who developed some of the advances in video microscopy, which increased contrast, and was at Woods Hole. And the other was that every day they would go out into the ocean and they would bring back squid. And squid have these peculiar neurons that are really, really enormous neurons. They're about 1 millimeter in diameter. And so they could literally dissect out one of these squid giant axons, take a tiny squeegee and squeegee out the cytoplasm and put it on a slide. And then they could take this, what at that time was newly developed microscopy, and look at the cytoplasm underneath the microscope with this video enhanced microscopy. It increased the contrast by a whole lot. And suddenly when they did that, they saw it looked like the LA freeway. There was traffic moving in both directions. And the question, of course, arose, what's moving them and what are they moving on? And that really started the experiments to sort out kinesin and dynein, and the fact that the movement was predominantly on microtubules. At that time, there were two schools of thought. Maybe it's the substrate is microtubule, maybe it's actin filaments. They couldn't sort that out from the microscopy, so they had to do electron microscopy and a lot of biochemistry to sort that out. But for now, what we have are these long-distance pathways, moving vesicles on microtubules over great stretches, up to 1 meter. We can think about the microtubules and the ATPase motors as being part of a long-distance transport system. And the way I analogize this is you can imagine a shipment of something you'd like to have in your house has come into the port of Tacoma. And they'll be on this big shipping containers. And those big shipping containers will be now taken off of the ship and put on railroad cars. And for us around here, those railroad cars may be delivered to Denver. And then in Denver, those packages will be taken off the railroad cars and repackaged and then put into boxes and brought to you by your local FedEx or UPS delivery man. So you have a long distance conveyor system on what we think of in our real world as being rail, and a local delivery system which we think of coming to your doorstep by truck. We can see a very similar arrangement here, where the long distance transport system is going to be on these microtubules, with vesicles and the motors being kinesin. But now what we remember what we're trying to do is we're trying to deliver a vesicle to the plasma membrane so that we can permit exocytosis. So it appears that much of this transport occurs in two phases. The first long distance phase is to use kinesin on microtubules, but there appears to be a local delivery system, and that local delivery system is one based on actin filaments. So what I want to do here is on this panel, I want to switch gears a little bit and now think about how actin monomer, G-actin, globular actin, polymerizes into an actin filament, filamentous actin, and how that filamentous actin can be used as a kind of roadway along which we can transport vesicles. Now, if we go back to the upper panel, and we imagine where we would find actin inside the cell, we find that the actin is almost entirely right here at the edge associated with the membrane. And this is called cortical actin, meaning it's at the cortex, it's the edge, it's a thin veneer right along the cytoplasmic side of the membrane. That's where we find most of the actin. Whereas the microtubules tend to be more toward the center of the cytoplasm. And you had a question.
Yeah, I was just curious, do the microtubules also move, or does the kinesin allow it to jump like between microtubules?
Okay, you have two questions here.
Yeah.
One question is, do the microtubules move? And the answer is yes. But they move at a much, much slower rate. They move at a rate that's more in the neighborhood of 1 to 2 millimeters a day, 0.5 to 2 millimeters a day, as opposed to 200 millimeters a day. So that's what we call slow exoplasmic transport. And your other question is, can vesicles move from one microtubule to the next? And yes, they'll get off one, they'll hop off one, probably wander about and get on another. Now, the distances between the microtubules in the cytoplasm is not very great, probably no more than maybe 100 nanometers at most. And so these guys can wander about, get their feet under them again, bind to a microtubule, and head off. Let's leave the slow exoplasmic transport till later. And we'll talk about that when we get to the nervous system. It's a very cool story, but let's wait that till later. Yes?
You said in reality the alpha helices of kinesin are intertwined. How do they step forward?
So the question here is if and I've erased them, if the two alpha helices are intertwined, how do they step forward? It looks like that alpha helix will unwind a bit and rewind. So every time you take a step, it sort of winds around, and then when it takes the other step, it unwinds. So left, right, wind, unwind. So it doesn't get tangled on itself. But it appears just at that linker region where you get these two rotating one around the other. And the two alpha helices appear to rotate around one another just at the linker region. Yes? Okay, so the part of the question here is about the disassembly of the microtubules. We're not going to disassemble them here. Once these microtubules are generated and they're in the cytoplasm, they're stable. They don't come apart. So we're not going to worry about here the story of assembly and disassembly. If you want to get into the story of assembly and disassembly, that becomes really important during cell division. And it's certainly true that microtubule assembly and disassembly is important for the microtubule to find the centromere on the chromosomes and pull the daughter chromosomes to two cells. That's all important for assembly and disassembly. That system works there. Although there's kinesin, which will move the chromatids along the microtubule in the same way, always toward the plus end. Right. But then, what was your other question? So dining will go the other direction. And that simply derives from the idea that if this is the plus end and this is the minus end, now the dining is going to be moving things in that direction, stepping along the microtubule just as the kinesin stepped, but going in the opposite way. So where kinesin is constricted, or restricted is a better word, restricted for moving from the negative end to the plus end, the dining is restricted in moving from the plus end to the minus end. So this now allows two-way traffic on one road. Anything else? Yes, in the back.
So once they reach their terminal, how do they? Does the body absorb them? How do they get back to the other end? You know what I mean? Like they're marching in opposite directions. They can't go in the opposite flow of traffic. What they're designed for.
So the question is, what? Let me rephrase that question. Say, what's the fate of the vesicles?
Correct.
Okay, and the fate of the vesicles, I'll try to answer that by saying these vesicles that are moving by kinesin in this direction.
No, I mean, what happens to kinesin and what happens?
Oh, what happens to the kinesin?
They can't go back to grab something else and march forward again.
Apparently, there are high concentrations of kinesin and dynein throughout the cell. And so it's not as though you have to make the kinesin here attached to a vesicle and it never leaves. It looks like the kinesin can come and go to some extent. Probably once it's attached to a vesicle, it doesn't come and go very much. But the kinesin appears to be able to come and go from the vesicle, so that you'll have a cloud of kinesin down here and a cloud of dynein. Then your question is, how do you regulate the attachment of one or the other to the vesicle so that you can move the vesicle one direction or the other? And that's not entirely clear.
No, my question is more that because you're going to end up with a buildup of more or less at the other end. Your variant, you know what I mean? Because they can only march one way. You know what I mean?
Right. Okay. So what?
You're going to end up with more on one end than the other.
Okay. So the way I have described this, you can envision something that's not quite right. You can imagine that vesicles move all the way down to the terminal, do something down here, get on to the microtubules and move back in the other direction, so that you have a consistent or constant number of vesicles that are moving back and forth.
Vesicles, dynein, right, moves from one end. Like up towards the head of the neuron, you think that it's full of dynein. Like,
right, but it's not. It's the dynein and the kinesin are sort of evenly distributed throughout the cytoplasm, waiting to attach to the vesicles.
are they made in either end? Are they made at both ends? Like,
They're only made up here. But then,
dynein will only march one direction. How does
no. There again, this comes back to the question that was asked here, which I didn't emphasize very much, and that is there are two kinds of transport. One of which is dedicated to vesicles moving, and the other is called a slow transport, and that's dedicated to proteins that are free in the cytoplasm. So molecules that are in the cytoplasm appear to move as a fabric, and at a slow exoplasmic rate, at least 100 times slower, more or less 100 times slower than fast exoplasmic transport. And what you're talking about are the molecules that were made on free ribosomes, incorporated into the fabric of the cytoplasm, and will move at a slow rate. But they'll be more or less evenly distributed in the cytoplasm through that. So you're not restricted to move the kinesin or dynein simply on this vesicular traffic. Okay? Okay, last few minutes. So now what we want to ask about is, how do we use this cortical actin to bring the vesicle in right up next to the membrane? So let's now go back a little bit and talk about actin-based transport. So again, I'll remind you that actin is polymerized from globular actin. So we have these individual, 40,000 molecular weight actins that can assemble one with the other. Now I'm going to offset this a little bit, because it turns out that in 3 dimensions, these actin filaments are twisted into a kind of helix. But be that as it may, each one of these circles is meant to be globular actin. And as with microtubules, there is a plus end and a minus end. And the terminology is the same. This is said to be the disassembly end, and this is said to be the assembly end. As with the microtubules, this gives us an orientation of the molecule. And as with the microtubules, there is a motor. But now the motor is another molecule. The motor, the actin, the actin-based motor is myosin. It too is an ATPase. It too has a foot, although in this case we often refer to it as a head. It too is a dimer. So that if we look at one of these molecules, there is this region of alpha helix, and then a foot, and a region of alpha helix, and then a foot. And the ability for this end of the molecule to bind to a vesicle. As before, the foot, or head, can bind ATP. And as before, it can convert ATP to ADP in inorganic phosphate. And the same pattern pertains. That is, here's the alpha helix, here's the foot, here's the alpha helix, and here's the foot. When this region of the molecule binds ATP, and it converts it to ADP and PI, when ADP and PI leave, then this molecule will step toward the plus end. So ADP, when ADP and PI leave, the myosin head steps toward the plus end of actin. So now, as we saw before, if this end of the molecule, if this non-actin binding region of the molecule is bound to a vesicle, we can now use the myosin as the motor and the actin as the substrate or the road. Now let's think of this in terms of the plasma membrane and the idea of using the cortical actin. So let's imagine here that this is the plasma membrane. And that there is this cortical actin. There are these short segments of actin filament. But they're oriented in such a way that this is the plus end and this is the minus end. And we can now imagine using the myosin molecules attached to a vesicle to transport this vesicle from the negative to the positive end, with the positive end attached to the cytoplasmic side of the plasma membrane, so that now we can deliver this vesicle right to the cytoplasmic side of the plasma membrane. This vesicle now is positioned so that through some other cell machinery, we can fuse the plasma membrane and the phospholipid bilayer of the vesicle so we can get exocytosis. In this way, then, we can imagine generating vesicles here, loading them with proteins and putting them on the microtubules to be transported by kinesin effectively down to the terminal. And then in the terminal region, the vesicles now can be offloaded from the microtubules, attached to the actin filament, and delivered right to the strategic site where the vesicle can nestle up against the plasma membrane and at that point release its contents to the outside world. And similarly, if we endocytose membrane, we can transport it in the other direction, and let's leave it there.